our work. Main problem in this domain is how to describe in single standard gene network processes that have different organization, function, and nature. So there is only one way to describe mathematical model of gene network. If you should do it by step by step, bottom-up or top-down methods. There are a lot of tools and some standards for mathematical model reconstruction, like Bionnet Gene, So Designer, ViewMail, and SBEmail, and SBW tools. But when you reconstruct the mathematical model of gene networks, you have to take into consideration the next aspects, specific of modeling process nature, clear interpretation of modeling objects, and model's reusability. And today I will tell you about SciBML language, this modeling language that evolves in the Institute of Cytology and Genetics. SciBML language was developed as the tool that can describe models from genetic till organism levels of hierarchical organization. SciBML is based on gene networks elementary subsystems methodology. So the simplest part of SciBML language is object, is the next structure. So this token is a model object identifier, and you can set necessary object properties as identifier attribute or set the compiler instruction in it. So the SciBML object identifier is a guarantee of correct objects comparison for two models if they both were constructed in same notation. So MGSmodeller is the editor tool for SciBML models. So how does it work? The gene network elementary subsystem in SciBML are represented a set of template blocks. It looks like this. Such block characterized the mathematical rule of variable relationship. The block has the variable sets and block-specific parameters on input. So at this table you can see some example of SciBML objects identifiers and notation. If you see, there are more than 70 blocks that can describe almost everything related to gene networks modeling. It looks like assembler, but is general for low-level language. We have created some compilers that may generate the SBEmail file, so you can work with these models in the tools that support SBEmail language. So how it works? The working principles I will demonstrate on new results that were obtained with MGSmodeller. More information about model you can read in Mironova paper in the Annals of Botany journal this year. I will tell about technical part of this work. This model describes auxin distribution in the root apical meristem. Auxin are a class of plant hormones or plant growth substances with some morphogen-like characteristics. Auxin have a cardinal role in coordination of many growth and behavioral processes in the plant life cycle and are essential for plant body development. Accumulating evidence suggests that the plant hormone auxin has the primary role in root apical meristem establishment and pattern. Auxin transport is driven by the asymmetric distribution of different auxin membrane carriers, including pin family efflux carriers. This model built on assumption that pin family efflux carriers are the main contribution to the formation of the auxin distribution pattern in the root. On the picture picture, you can see the family proteins localization on cell surface. Auxin transport and pin family proteins, auxin transport and pin family protein synthesis and degradation were described as the elementary subsystems, like about 14, if I'm not mistaken. For modeling purpose, 2D cell structure with 4 columns and 20 rows, as you can see, cells layout was selected because root has the central symmetric. The compilation script was created that assigned all processes with the cells layer. After compilation, we obtained model with 240 variables and 39 parameters. Calculation time takes from 20 minutes to a few hours from high performance cluster. As I said, the MGSmodeller tool includes calculation routine GIR method, but it also has option to save the mathematical model in MATLAB mathematical step-list formats for testing and for sharing these models. It works on high-performance cluster. So to use the preferences of high-performance calculation, we developed the model calculation method with initial conditions and parameters varying. We have used this method to solve the parameter explanation problem. As a result, we obtained thousands of calculations results. So main equation, how to analyze such amount of data. Even the single result vector is not too simple to analyze, as you can see. So what to do with it? As I mentioned, we have the structure identifier. Attributes of identifier in this case represent the object name, type of the object, and localization in cells layout. So all we need is to sort and rearrangement of variables. Main idea that we cannot predict the structure of models that will be explored with our tool, but we can give a comfortable identifier format that could be processed by other rearrangement subprograms. And we have done some utilities. After result preprocessing, we obtain that data that are suitable for visualization utilities like GNU-plot or MATLAB. So we can see the variable values spread on the cell structure and could easily compare it with the experimental result. And we can even obtain the variable spread in dynamic as animation. This MATLAB, for example. So, as I said, you can get more information about the model